So welcome to The Mystery in Our Genes, Why We Are Who We Are workshop today with Ed Cloza, genetics consultant at the New England Regional Genetics Network. All of our workshops, including previous ones, are recorded and can be watched on our VFN YouTube channel. The link is on our website, which is vermontfamilynetwork.org. Everyone who registered for today's workshop will receive a follow-up email with a link to this recording and a PDF of the presentation with live links. Vermont Family Network has been serving families of children with special health needs and disabilities and the professionals who serve them for more than 30 years. We are the federally designated Parent Training and Information Center, the Family to Family Health Information Center, and the Vermont chapter of both Family Voices and Parent to Parent USA. Today's workshop is being recorded, so please keep that in mind as you share information about your situation in the chat. These sessions will include live captions. If you want to turn them off, click on Live Transcript, then Hide Subtitle. If you are comfortable, you are welcome to turn on your camera. You can ask questions using the chat box, which you'll see at the bottom of your screen. Feel free to share information or links to resources that you found helpful in the chat as well. We know that we can learn a lot from each other. I'm very excited to introduce you to Ed Cloza. Thank you so much for joining us today. Ed, the floor is yours.
Well, Jamie, thank you very much. And it's a pleasure to be here talking to folks in Vermont. In talking about genetics, I love talking about genetics. I've been in the genetics business for over 40 years. I'm a genetic counselor by training. And I love to take the opportunity to help inform people about this, to me, very fascinating subject. And it's so complex, but I hope to be able to kind of reveal some of the secrets of genetics so that you have a better understanding of it. I'm going to switch over to my presentation now. Yeah, the mystery in our genes. And it really kind of is a mystery because until relatively recently, nobody knew very much about genetics. And there's still a lot more to learn despite the fact that we've come an awful long way. What I hope to do today, here we go. Number one is to help you understand how this simple chemical code is used to write an instruction book of life, a book that not only can dictate how human beings are built and grow and stay healthy, but the same code is used for other animals, for plants, for bacteria, everything. And it's really fascinating to me how this kind of all works. I hope to have you understand why some features of parents are passed down to their children and some aren't. And finally, to explore why and how some genetic changes happen and how those changes might impact development and health for better or for worse. I'm here on behalf of the New England Regional Genetics Network, which is one of 7 regional networks funded by the federal government. And its interest, all of these networks' interests are to help identify within their own geographical regions folks who could use connections to genetic services and resources and help to facilitate those connections. Genetics is complex. It's sometimes not easily accessible by everyone. Some folks and some physicians and clinicians don't realize where the resources are. We hope to kind of make it easier to make the connections. In New England, the project is housed at the University of New Hampshire in Durham, and it's directed by Dr. Kim Phillips. But if you were to get in touch with Nergen, it's more likely you'll be talking to Karen Boley, who is the person that kind of makes the trains run on time. So why do I think it's important for people to understand genetics? Well, most people haven't had much exposure to genetics except maybe in high school biology as sophomores. And for someone like me, so much has changed, and every day there's new information coming out. But genetics is not something that's alien to anyone because it's pervasive in the media. I mean, shows like CSI, um, the do-it-yourself at take home DNA testing kits to help identify where your roots are and help bring you closer to other members and connect your family. It's a big part of life, and it in fact is a big part of every aspect of medicine. And more connections are being made every day. But if you understand genetics, it might help you understand the cause of a child's condition. It might help you understand why certain testing, treatment, or management is being recommended. It might help you to expect more about what to expect from a child's progress and why. And then finally, why in some cases other family members may benefit from the knowledge that you may have about a child's condition. The bottom line is, as with most information, the more you understand, the more power you have, the more integrated you can become in your child's care. So now I want to take a few minutes to kind of help you understand. For many of you may have only had their sophomore biology genetics exposure, but some of you may have for one reason or another kind of kept up with genetics a little bit more. And I'm certainly not intending to make anyone a genetics expert here. If you wanted to be a genetics expert, you could have gone into the field, but I do want to kind of help get us all on the same page with regard to basic understanding of how you can take something like DNA, as simple and as complex it is, and turn that into a human being, and how changes in DNA can cause problems. Rather than talk about molecules and structure of DNA, which I don't think would be quite helpful, I like to imagine this whole human genome thing as a kind of a metaphor for that would be like having a set of instruction books. And this set of instruction books has every detail in it to tell you how to make an entire human being. Interesting thing about this instruction books is that the words are very few. There's only about 22 different words in this whole book. And in fact, each word is very small. Each word is only 3 letters long. And what's even more interesting to me is that the alphabet only uses 4 letters. So you've got an instruction book for something as complex as a human being. And that instruction book is written with an alphabet of only 4 letters, written in words that are only 3 letters long, and there's only 22 different words. Yet everything that we are physiologically is created by stringing those different words together in extremely complex sentences. But it's a simple way to start, and it's a very complex organism that we have. Now, not only do you have one set of instructions, you have 2 sets of instructions. Every one of us does. And we get one set from our mother, and we inherit one set of these instructions from our father. And these instruction books come in volumes of 23. So we're getting 23 books from mom, 23 books from dad. Everything you need to know how to make a person is in there. And these books are pretty much similar from mother and dad. Not exactly, but close enough. So let's say you cut yourself and your body has to make some new skin cells. Well, what happens is you open up that book to the volume that talks about making skin cells. You go to the page, and the instructions are read for making skin cells. Reads the instructions from the mother, reads the instructions from the father, uses a combination of both of those, and you get new skin cells. Boom, and the scar heals up, and you're done. And this goes on millions of times during the day, not for skin cells necessarily, but you're always making new cells. You're making new hormones. You're making hair, your skin is growing, your fingernails growing, everything is growing, and it's under the direction of these instruction books. The interesting thing is every cell in your body, except the red blood cells, has this entire set of instructions. But your skin cells, despite the fact that they have the ability to make brain cells and liver cells and insulin, your skin cells only make skin cells. And they don't make it all the time. They only make it when it's needed. So the instruction books are smart enough to only open when they're needed to be opened, where they're needed, and the appropriate material is made if everything goes correctly. Now, let's switch from talking about this metaphor of the encyclopedias or the instruction books to the real human situation. So if you were to take a skin cell and examine it under certain laboratory conditions, what you'd find is these structures inside the nucleus. The nucleus, as you remember from high school biology, is kind of the command center of the cell. And inside are these structures. They don't look this way. They're not colored this way. They're not even shaped this way typically. But in the laboratory, you can get them to look this way. And if you took a pair of scissors and cut these out, one thing you'd notice is that they do match up in pairs. This kind of yellow one matches with this one, this orange one and this orange one, this purple one and this purple one. And you could actually put them on a sheet of paper and match them up according to how big they are, kind of what shape they are, and in this case, what color they are, and find there are your 23 volumes of the instruction books. One of each came from mother, one of each came from father. This set down here where it says X, these are the sex chromosomes. This person has 2 X chromosomes, that's a female. If it had 1 X and 1 Y, that person would be a male. But these are the entire catalog of making a human being. You can't get a real good idea from looking at this picture what these structures are made of, what they look like. This one tells you a little bit more. This is an electron micrograph. And to me, they look like fuzzy bedroom slippers. But what I hope you might be able to tell is that they are in fact a kind of a thread-like material, and there is a beginning and there is an end, and the rest of it is one big blob of like yarn, all bunched together, very, very tightly compressed in this illustration, in this image. But you can grab one end and you grab the other end and stretch it out. If you took one skin cell and took the DNA from one skin cell, stretched it out, and laid it end to end, that string of DNA would be about 6 feet long. So one cell has a tremendous amount of DNA in it, all scrunched up in these tiny microscopic chromosomes. So just to remind you, there's your cell, there's the nucleus, there's the chromosomes. And if you were to stretch the chromosome out, you'd find that's all made up of a long string of DNA, deoxyribonucleic acid. But the thing is, you've heard of DNA testing, etc. And one particular stretch of DNA that's responsible for making a protein, like keratin in the skin or nail, is a gene. So you can assign a stretch of DNA to say this makes insulin, or helps to make insulin, or helps to make a white blood cell, or helps to make a brain cell. And these are the words that are being used. And if you took this DNA and stretched it out, you'd find that it's got this shape that you should be familiar with because its logos are used by perfume makers and everybody else who wants to try and make their image look kind of hip. This is the double helix that they say DNA is made out of. And these are the 4 letters, T, G, A, C. There are no more letters than T, G, A, and C used in DNA, in the gene. And if you look at any group of 3 of them, TGA, that's the word. That's the word that is used to help string a protein together, like insulin, for example. And it's as simple as that. It's as complex as that. Well, how do genes make us different? Well, first of all, if we were to look at the genetic material, my genetic material, and compare it to the genetic material of anyone else on this presentation, we'd find that 99.9% of the material, the words, are exactly the same, no different. And that when you think of the difference between me and Jamie, or Jamie and anyone else on this presentation, much of that is caused only by the 0.1% of the instructions in our DNA that are different between us. We're far more alike than we are different as humans. But there are changes. And again, there's so much DNA. If you took all the DNA from one person, stretched it out like I suggested, that string would stretch from the earth to the sun and back 10 times. So yeah, only 0.1% of our instructions of our DNA are different, but there's so much DNA that there are ample opportunities for changes to occur and make us different. Let's talk for a minute about genetic changes. Well, sometimes these changes can occur at the chromosomal level. So here's another picture of chromosome, right? Remember, you're getting one from mom, one from dad. These are colored a little bit differently, but they're the same. There's chromosome 1, which is the biggest one, and all the way down to 22, which is the smallest. This happens to be a male. There's the X, there's the Y. One from mom, one from dad, one from mom, one from dad. What happened here? Chromosome 21, instead of 2, there are 3. It's perfectly normal. It's either mom's or dad's that it came down with. But there are 3 chromosomes instead of 2. So when your body needs to make something that's coded for on chromosome 21, instead of opening up 2 copies of that instruction book, it opens up 3. And it makes more material. It uses an extra set of instructions that really weren't intended to be there. It's reading 3 copies instead of 2. And 3 copies of chromosome 21 result in what we call Down syndrome. Pretty common. We all know it. We've seen it. It's just the addition of an extra chromosome. Normal chromosome, nothing wrong with it. But the contribution of that extra material leads to the typical features. So these can be seen in a laboratory under a microscope. These can be seen. You can have extra chromosomes, you can have missing chromosomes. Sometimes a whole chunk will be gone. Sometimes a chunk will be switched around. One piece of a chromosome will wind up on another chromosome where it shouldn't be. So there are many ways to have these changes occur when pieces of chromosomes are altered. But more of these changes occur in a way that can't be visualized under a microscope. They happen when, as a person grows and has to make new cells, the DNA can actually, as it's being copied, that's when errors can occur. You can imagine when you're copying anything, changes can occur. Letters in the DNA can be different. Words can be different. And there are plenty of opportunities for these changes to happen. And most of the time, these changes happen as we get older and as the copying mechanism starts to kind of break down a little bit, and you get these changes. But sometimes things in the environment can do it too. If you stay out in the sun too long, the sunlight, the ultraviolet light can alter the DNA in your patch of skin, and you can get skin cancer. You can get lung cancer from smoking cigarettes because the carcinogens change the DNA in your lung cells, etc. But most of these happen naturally as we get older. This is nature putting these changes in. It's like mutating COVID virus. It causes these changes in an effort to help improve the population. Well, how can these changes help improve the population? Because sometimes these changes can be good. You can get genetic changes in your DNA that help you live longer, that help you fight off infection a little bit better, that help you make it easier for you to have kids. Sometimes these changes don't make a difference. They can be neutral. Doesn't give you a disadvantage, but it doesn't give you an advantage either. The ones that we're concerned about are the changes that are harmful, that affect lifespan, that affect health, that affect the ability to have kids and pass your genetic material on. And again, sometimes these changes affect only certain cells, but sometimes they can be passed on from parent to child. And those are the ones that we're more concerned about. Now, let me show you a little bit about how these changes may happen. These are called leading mutations. So again, we're talking about getting one set of instructions from mom, one from dad. And let's switch our analogy a little bit now from an instruction book to a recipe book, which is kind of the same. So let's say you're making something and the instruction you get from your mother's set of recipes is use one egg. That's what you're supposed to have. You open up the instruction from your father, it says use one egg. Okay, you know what to do. You use one egg because your mother's book says to. You use one egg that your father's book says to, and you add 2 eggs to your recipe. What if your mother's instruction is okay, but your father's instruction has a change in it? And now it says use 2 eggs. It's just like that. Well, I think a lot of people, if they saw that, they would say, oh, it's supposed to say use 2 eggs. But I get it. I understand what they want me to do. They want me to use 1 egg from mama, use 2 eggs from my father's instructions, and put 3 eggs in. I was really only supposed to have 2. Now you've changed the recipe, you've changed the instruction. Excuse me. Well, maybe 3 eggs in this recipe is an improvement. Maybe it's a disaster. Maybe it makes no difference at all. You can't taste the difference. But it's been changed. Now, what happens here? Mother's instruction is correct, but somehow a W got inserted in front of the father's instruction. And remember, we're looking at 3 letters at a time in the DNA alphabet. Now it says, was ion ig. It's nonsense. It's gibberish. Doesn't make any sense. Well, you know you're supposed to use 1 egg. But what the heck are they telling you to do here? You don't know. Oh, okay, I'll make 1 egg. Again, this recipe, if it has 1 egg instead of 2, might be better, might be worse, might not make any difference. It might be inedible. Big problem comes in, and when both mother's message and father's message have mutations, have errors, the same error. So now you get to this part of the recipe. What do? I don't know what I'm supposed to do. This doesn't make any sense to me. I'm not putting any eggs in. So number 1, the material you're making, the product that you're making is going to be defective because it doesn't have the right ingredients in it. Or perhaps you might just not even make anything because you say this is a poorly written recipe I'm not going to make it. So you don't add any eggs, or you don't make the thing at all. If these were instructions for making insulin, and you don't have the right material in that gene to make insulin, the insulin is going to be improperly created, and it could be either non-functional or partially functional, or maybe no insulin is made at all. This is how these changes that can occur on a molecular level. You can't see these under a microscope. But these tiny, tiny changes can be enough to have a very significant impact on health. Now, the truth is that every one of us, me, Jamie, and everyone on, has about 400 of these genetic changes in our gene system. Some of them could be harmful, but they're not harmful unless they match up with other harmful changes, like in that last example. Some of them could actually be beneficial and give us a little advantage, make us a little healthier, a little more resistant to infection, make us process our food a little bit better and get more nutrients out of our food. But these changes, which we all have, we can pass some of these down to our kids. But among those 400 changes, about 60 of them are changes that we did not get from our parents. They're brand new to us. And our children have about 60 changes that they don't get from us. They're brand new to them. So again, nature is changing things up. It's shuffling the deck. It's trying to make sure that there's enough variation in the population that if some big disaster happens, the population will continue to survive. Genetic testing can sometimes find these changes. Because the genome is huge. There's like 3 billion base pairs, letters, and testing now can find every single one of them, but it's still a little difficult to figure out what that all means. So for something like Fragile X, there's a gene called the FMR1 gene on the X chromosome that I showed you, and you can do testing to find out if someone has Fragile X. You can do testing to find out if someone doesn't have Fragile X, but could have a child with Fragile X, or could have symptoms of something related to Fragile X as they get older. There's a tremor that can occur to folks who don't have Fragile X, but who can develop this stuff later on. And women can get primary ovarian insufficiency. So this testing can find many of these genetic problems, but sometimes testing will find the changes, but they won't. The clinicians can't figure out whether the change. So if they test a child and say, "Ooh, we have been able to identify this variation, this change in this child's DNA," well, does that mean it's responsible for the child's condition? Don't know. Let's look at mother's and father's DNA. So that's when they may come and say, we need samples, skin samples, or cheek brush samples, or blood samples from mother and father. And if they find that same change in mother or father, but mother or father are not affected, then they'll say, well, that's just one of these changes. It doesn't make any difference. If they don't find that change in mother or father, they'll say, aha, this change was associated with this condition. We still can't say for sure that it's responsible, but it gives us a lead. So genetic testing can be very helpful in finding out what may be causing a problem with any individual, but it doesn't guarantee that genetic testing doesn't guarantee that it's going to come up with the right solution. Well, how do these changes get passed down to our children? So very briefly, among the cells in a male's body are cells that produce sperm. And these cells, the sperm-producing cells, like every other cell, has 46 chromosomes. But what happens, this is a specialized cell, when it creates sperm, it only gives half of the genetic material. It gives one of every pair to every sperm. So, and which one goes, or which two go, which chromosomes go into that sperm it's a toss-up. It's a flip of a coin every time. Same thing for a woman's cells that make eggs. One big difference, when a woman is born, the minute she's born, she's got about 100 million eggs. That's all the eggs she's going to have. Oh, excuse me again. The minute she's born, she starts losing eggs. Man, on the other hand, produces sperm frequently. About 1,500 sperm cells a second can get produced. So the opportunities, again, for errors to occur, a little bit different between men and women. But bottom line is that every sperm is going to have 23 chromosomes. Every egg is going to have 23 chromosomes. And when the sperm and the egg get together at conception, you're building an embryo, developing baby that's got 46. And there is ample opportunity for changes to happen, so that all of these eggs that a woman has are all different. And all of the sperm that a man produces are all different. There are no two alike. Because not only do they have opportunities for the chromosomes to get mixed and matched, but even within chromosomes, things match around. Nature is very carefully has this kind of a system where it tries to maximize the variability, to make people as different as they can be, for better or for worse. Well, when are genetic problems found? Well, they often can be found before birth. You can do blood testing to look at proteins that mother's making that can give a hint as to what's going on with the developing baby. You can actually look in mother's blood for DNA from the developing baby, and it'll give you a clue as to whether or not the number of chromosomes is what it should be, 2, or whether it's this one chromosome, there's only 1 or 3 or whatever, whole different sets. You can also sample the amniotic fluid from around the baby, because the baby's always shedding blood cells anyway, or shedding cells anyway, so that you can get a direct look at the baby, developing baby's chromosomes and DNA. You can look at material from the placenta as well. But prenatal testing is something that you have to give careful consideration to, and have to give some consideration to how you're going to manage the information that you get. Very often, though, problems can be found at birth through newborn screening or by exam, the typical exam that newborns get, minutes after birth. Every state in the country has laws regulating that newborn babies have to be tested for a handful, not necessarily a handful, for a series of genetic conditions. In Vermont, that's 33 genetic conditions, plus they do a hearing screen, and then they also put a little blood oxygen monitor on the baby's finger to look and see if there are any heart problems. After August 10th, you can, if you want more information, you can see the brief video on the Vermont Family Network YouTube channel. I recommend you take a look at that if you want more information about newborn screening. Every state is different. Some states test for more, some states test for fewer. But the reason that this testing is done at birth or within a couple of days after birth is because for many of these conditions, in which the baby's body is not making a needed enzyme or a needed protein, or maybe too much, if you catch it early, before while the baby's in the womb, the mother's body is kind of taking care of the baby's nutritional needs. And but once the baby's born and separated from mom, it's up to the baby's genetic system, genes and genetic information to make everything that's needed. And if there's errors there, and there's too much or too little of a protein or enzyme being made, you can step in and provide medication, you can change the food, you can take all kinds of steps to prevent problems that would otherwise occur if no testing was done. So that's why it's a public health issue, and it has saved millions and millions of babies from having these conditions, which were used to be pretty rampant back in the old days. A lot of conditions are found in childhood because their kids are not sitting up when they're supposed to, not smiling, not talking, not walking, maybe when they get to school. I'll also remind you that there's a presentation on helping kids as they get back to school, settle back to school. It's going to be part of this summer series that's being presented by the Vermont Family Network, so catch that. Sometimes these issues are not found until adulthood. When you do a mammogram, you find breast cancer, and much of that is caused by genes that predispose you to breast cancer or colon cancer. You can get genes that can cause very, very high cholesterol levels, even in young kids, or it can cause the inability to process iron that you eat in your food. Everyone wants to eat iron-rich foods, except if you can't break it down, that iron can wind up in your brain, it can wind up in your liver, it can wind up in your joints. Plenty of opportunity for things to kind of go wrong. Well, what do you look for if you're looking for a genetic condition? Certainly a family history. If there are relatives in many generations that are affected, that's a huge sign that this is a genetic disorder. Groups of conditions present at birth, a syndrome, not just one little problem, but 5 or 6 things that are consistent, that can be sign of a genetic syndrome. Early or unusual symptoms of common conditions. If you look at a 12-year-old kid's cholesterol level and it's 200 or 300, that's not right. That shouldn't be there. And that could mean that he's having trouble metabolizing cholesterol. Neurodevelopmental delays, kids not hitting their milestone, very common. Someone who has rare tumors or tons of tumors, that shouldn't happen, but will happen if there are genetic changes in the signals that tell a cell to stop making new cells or to start making new cells. There are cancer genes that can trigger tons of tumors. Laboratory results that shouldn't be there in a healthy individual. You get very high iron levels. Think of, hemochromatosis, which is a common dominant form. Passes down through families, and it can put your health at risk. The treatment for that, give blood. The more times you can give blood, you're dumping the iron. Most people that need blood could use the extra iron. So it's a very simple solution to a very complex molecular genetic problem. Kind of wrapping up here, the resources that are available for folks who have kids with genetic conditions. If you've got a child with special needs and you're worried that it might be genetic, make sure that someone's looking after you, whether that be the child's primary care physician, a visiting nurse, healthcare provider, even a family member or a friend who can help you connect to the appropriate care. That's great. And if you would like more information for yourself, for your family, or even for your doctor, look for it, and look for it in reliable places like the National Family Center, like the New England the New England Regional Genetics Network website, which are listed here. And specifically, the Vermont Family Network has a presentation coming up on genetic resources in Vermont, and I would highly suggest you take a look at that as well. So that is a little insight, hopefully, into the mystery in our genes. I'm sure it's raised a lot of questions. I hope it didn't confuse you too much. Hope it was helpful. And if you do have any questions, I'll be happy to take them now through Jamie. You can send them in on the chat and she'll kind of pass them on to me. You can also get in touch at info.negenetics@unh.edu. So thanks very much for your attention.
Thank you, Ed. So we do have a few minutes for questions. Feel free to put any in the chat, or if you want to, actually, Rachel, I don't know if people can come off mute, but if you want to put them in the chat. So one question that has come up, what about mental health issues?
Yeah, mental health. I mean, there was a time in which if you asked, what can genetics affect? Well, yeah, we knew it could affect what we used to call birth defects, things like that. But not much thought was given to things like mental health issues, alcoholism, dementia. And we realize now that nothing, even, we'd say, oh, if you fall off a ladder, that's got nothing to do with genetics. Well, not necessarily true, because if you fall off a ladder because your equilibrium is off, it could have a genetic component to it. If you fall off your ladder because your vision is bad or your coordination is bad, everything has a genetic component to it. And clearly mental health does as well. The problem with things like mental health is that genetics does not work in a vacuum. It's not just genetics. Genetics and environment work very, very closely together. So two twins, identical twins, have this exact same genomes and genetic makeup, but they can have different behaviors because of the environment that they grow up in. They can have different behaviors because of the food they eat and the medications they take, or the vitamin supplements that they take. Genetics works in conjunction with environment, and mental health has a genetic component, but there are no simple tests that you can say, I found the gene for someone's bipolar disorder, or I found the gene for schizophrenia, or I found the gene for depression. Most of the time with these issues, there are many, many genes that work with each other. They work with the environment to exacerbate or kind of maybe make it a little bit milder. But it's a huge problem, and that's why we know that medication will work in many cases with mental health issues, because the medication is changing the biochemistry in the brain, and the biochemistry in the brain is largely caused and created by the genes. So yeah, that's clearly mental health issues has a genetic component. Unfortunately, not clear enough that we can say, I can track this, I can follow this, I can fix this.
We have another question, Ed. The comment reads, that was so interesting, thank you. Is it hard to get in to see a geneticist? We had testing done when my daughter was very little and it was inconclusive, but now that she is an adult, we have a clearer picture of her challenges. Do you think it would be helpful to pursue testing again?
Oh, great question. Testing has come a long, long way over the past 10 years, and it's going to go further in the next 5 years. Uh, the ability to link genetic variants, genetic changes with genetic conditions is improving all of the time because more people are getting tested. This information goes into databases that are funded by the governments, and it's all this information is put together not only within one country, but worldwide, because some of these conditions, genetic conditions, are very rare. There may only be, 100 people in the world that have been diagnosed with it. But if you can say, look, we have a common genetic variant that we're finding among these 100 people, it's got to be a big clue. And then that way, when another person turns up with this thing, you're kind of on the road at least to getting a diagnosis. So the answer to the question, is it worth looking into testing again? My suggestion would be get back in touch with the people that did the initial testing and ask them that question, because they may say, in this case, nothing new or nothing significant has come up, but there are a couple of studies underway. We expect there to be more information in a year or two. Or they might say, yeah, there are a couple of labs that seem to have some new information that could be very, very useful in your case. It's in general, it's always wise to go back to the people that did the original testing and say, what do you think? Is there, you got anything more to tell me? Because sometimes what you tell them and say, oh, my daughter now, these are the challenges she's facing. These are the things that she's overcome. Here's what we've been able to figure out. That may give them additional clues as to say, we were looking kind of the wrong direction the first time. Let's try this other route.
Great, thank you. So I don't see any other questions in the chat. So I think that's it. Any final words?
I see one up there. Is there a specific?
Okay, yep. Is there a specific resource you might recommend for finding early developmental information on children with specific deletions? I am an early educator and feel it isn't fair to these children or families to expect them to align with typical milestone achievements.
Oh, great point. Again, if there's enough information, if this is so rare that you get 100 people spread across the world, it's very hard to pull together information to say this is what we can expect. But deletions can be tricky because there are known syndromes attributed to deletions, and they're pretty much all, I don't want to say all the same, but they're well characterized and you kind of know what to expect. Sometimes deletions, and that's where a chunk of chromosome or a particular piece of a gene is missing, that if someone's got a deletion of 500 base pairs, 500 letters of the alphabet, someone else has a deletion of 525, those extra 25 may or may not be significant, and they may or may not result in different challenges for that kitty. But it's many families who have kids with specific deletions have come together and say, hey, look, we all have kids with, a 4p minus or something, missing a small piece of the small arm of chromosome 4. Let's all get together and we'll talk about how, you handle this problem, how you handle that problem. And so if you can look up specifically for that deletion, family groups, family-led organizations, you can compare instead of saying, well, my kid not doing what every other kid in school is doing at the same time, but my kid's doing fine if you look at all the other kids with this particular deletion. It's right smack dab in the middle. This is where they're doing exactly where they're exactly where they should be for the condition that they have. So if you've got specific conditions, I would go to something like the National Organization of Rare Diseases, NORD. They may help. There are a lot of good websites that you can find on the internet. There are some that I'm not sure I would trust, but if it's something that's run by the government, by NIH, by a reputable organization like the Mayo Clinic has great stuff like that, I would look there. And again, you can go back to the physician or geneticist that made the diagnosis and say, are you aware of any particular helpful websites? The New England Regional Genetics Network has a website called GEMS, Genetic Education, something with school success, materials for school success. And we've got about 40 different conditions that we have very detailed information on, all targeted toward helping these kids do well in school, helping the teachers understand what the limitations are, helping the other students understand, why is this kid different, helping the teachers understand, if we go on a field trip, this is what I can expect. This is what I can expect for nutrition. This is what I can expect for absences, etc. All designed to help focus on making sure that the kid with the condition can maximize their potential so that they can do as best as they can in school.
I did put the GEMS website as well as the NORD website in the chat. So a kind of a follow-up was, I have a family who has a mom with the same deletion as the child. Should I expect him to grow up to be similar in his abilities to mom's? His is 17q11.2. So it sounds like I can look it up, but do you have any thoughts on that specific condition?
Not on that specific condition, but it's interesting that mom and child have the same condition. And in general, the answer is yeah. If the child has inherited that from mom, and it hasn't expanded, it hasn't gotten any bigger, that deletion, then you could probably expect there to be pretty similar development. So yeah, if mom's doing fine, then I would think the kid would be doing fine. Now, having said that, I mean, there are other things that may be going on, but yeah, in general, if you only saw that in the child and didn't see it in a parent, then you'd say, "Who? I don't know what's going to happen here." But if you see it in mom and it's not doing her a lot of harm, that's a good sign.
She offers that mom has significant ways.
Well, then you kind of you can expect it, but the individual care of that child and the mother should be in the hands of someone who knows how to work with those types of developmental delays. And my guess is, when the mother was growing up, the type of resources that were available to her were not as good as what's available to a child now. That's a general statement, but things seem to be better now in terms of resources and understanding and expectations than they were, 10 years ago or so.
Great. Well, we are just about up on time. So I wanted to thank you again, Ed, for today's informative workshop. All of the participants will see a link to the evaluation in the chat. If you could please take a few minutes to complete it so we can make improvements and fulfill our grant requirements, we would greatly appreciate it. So today's workshop is one of 4, as Ed had mentioned, in our summer genetics series. The remaining 3 workshops will be pre-recorded, and we will post all 4 on our website later this month. We have many more interesting workshops coming up, and you can find details about those on our homepage of our website, which is vermontfamilynetwork.org, along with the links to the previously recorded ones. We have tons of information on our website, and getting connected with us is easy. You can follow us on Facebook or join our closed Facebook page if you are a parent. The name of the closed Facebook page is Vermont Families of Children with Special Healthcare Needs. You can also sign up for our monthly e-news or check out our YouTube channel. So we are here to help. Please contact us by phone or email. And thank you everyone for joining us. And thank you again, Ed. This workshop was excellent. So thank you.
Thank you. It's a pleasure.